Hi everyone, my name is Aurelien Pelissier. I'm a PhD student in IBM Research on ETH Zurich. And today I will present modeling of the immune system, a multi-sketch challenge, with a special focus on the germinal center. So first, the motivation for our work is, our aim is to do in silico immunization, which could be very useful, for example, to develop immunotherapy for cancer or optimizing vaccine strategies. And such model would be very efficient compared to in vivo immunization. Typically, in mice, it takes a month to test a vaccine, whereas in a computer, it takes only a few hours of simulation. Of course, such objective is very challenging, and today we focus on a specific part of this immune response, which is the germinal center. So germinal centers are found in the leaf node here. They form in response to the presence of foreign antigen, and they build a response typically in a few weeks. So such germinal center has many different factors to consider, because first there is hundreds of them and they interact with each other by exchanging cells, and each of them contains around thousands of cells. And of course you have to consider the molecular binding between the antigen and the B cell receptor. So in more details, the germinal center is a place where B cells, here in red, they undergo a Darwinian selection process. So first they divide here in the dark zone, and then they migrate to the light zone where they acquire antigen and the follicular dendritic cells. And once they acquire antigen, they interact with the T cells here, the T cell in red. Depending on the response of the T cells, they can either leave the germinal center as a memory cell or plasma cell, or they can recirculate here for more proliferation. And finally, if they didn't acquire enough antigen, they cannot bind to the T cells and they undergo apoptosis. Such germinal center is a dynamic system, so at the beginning of the response it's very small and it has a huge increase in number of cells and then start decreasing again. So how do we model this in our in silico model? So what we do is we prepare a set of reaction, 11 in our case, and it looks like a chemical reaction. So you have some reactant here. CB is like centroblast, which is in the dark zone, centrocyte in the light zone, and each reaction is associated to reaction rate. So it's very similar to a chemical reaction, and we simulate it with a Gillespie algorithm, which is very widely used for biochemical reaction. And then for the genetic events, so we only consider a few mechanisms. The first one is mutation. So every time a cell divides, they will undergo mutation. And the mutation has three possible outcomes. It can either do nothing, which would be a silent mutation, or it can cause apoptosis because the B cell receptor is unproductive and the cell will die of apoptosis. And finally, the mutation can change the affinity of the cell, and this is the important part. And also the division of the B cell is regulated by MYC, which is upregulated by the T cells. And finally, the last thing we have to consider is the antigen binding. So B cell receptor and the antigens, if you look at the 3D molecular structure, it's very complex. But in our case, we use a very simple model, which is the Hamming distance. So we just compare the nucleotide sequence. If the letter is the same, then the affinity is increased by 1. And if the letter is different, the affinity is 0. So as an example here, we have an affinity of 0.6. And here is our result. So we fit all of model parameters with experimental data from the B cell count of the cell count of B cell, or the number of mutations per 10 days, the death rate. And with all of this, we can obtain a realistic model, and especially we can look at the affinity maturation. So here it's clear that the average affinity of the B cell in the germinal center increase with time. And because we keep track of the nucleotide of the B cell receptor, we can visualize a phylogenetic tree, lineage tree. And finally, the last thing we can do with our model is understanding the emergence of clonal bursts. So a clone is a cell that descends from the same founder cell. So at the beginning of the germinal center response, you have few clones that enter the germinal center, and then they proliferate, which they create a clonal family. And in this specific example, the brown clone had a high affinity jump by chance, and few days later, it translated into a clonal dominance of this clone. So this is what we could see with our model, but still there is still many things to do because we focused mainly on the cell interaction, where we did some very strong assumptions for the molecular binding and the gene regulation. So further work will have to consider also a more precise 3D structure of the molecular binding. And of course, we are very interested to see what happens when we consider multiple germinal centers at once, where the germinal centers exchange cells with each other and are specialized to different antigens. So that was a summary of my poster, and thank you for your attention.